It's lectern again. So I'm not going to be able to do quicker questions today, unfortunately, or fortunately for you. So what we're going to begin our next arc doing is looking at some applications of pathogenesis, bacterial pathogenesis, and really giving you, I hope, an important glimpse into how do we know some of the things that we know. So I've tried to give you some ideas of like, here's some experiments that were done, but in terms of assessing how nasty a particular pathogen is, and so on. But there's a whole lot now that essentially, I would say the last 10 years, we have learned more about pathogens than we have for the last 100 plus. And one of the reasons is that we're able to compare pathogens to each other. And so the last paper we discussed, being able to distinctively separate salmonellas from different places out into different subspecies, and that knowing that these subspecies are associated with particular hosts, and so on. A large part of those differentiations that we can make are driven by this, that we can access the genomic information of culturable and now unculturable pathogens and understand a lot about what they do to be pathogens through comparison to other model pathogen systems. And so that's kind of the big picture that I want to try and drive home today. And so when we're talking about genomics, one of the things that I think is important to start with is just kind of getting everyone's minds back into what is a prokaryotic genome. What am I talking about when I'm talking about genomes? And how do we study prokaryotic genomes? In particular, how do we study pathogen genomes? And how do we connect specific genes in pathogens to pathogenic traits? Okay. So, if we were to kind of rewind the clock and go back to microbiology, the characteristics of the prokaryotic genome is they're comprised of DNA. They're composed of chromosomes. So these chromosomes are circular. Now there are some linear chromosome bacteria. I think one of your pathogens for sure has a linear chromosome. And there's usually one large chromosome that is circular. So we draw these circular maps of chromosomes in contrast to what you might see for the eukaryotic system, where these chromosomes are linear. You can see linear maps of chromosomes. And there are some pathogens that have multiple chromosomes. So Vibrio cholera, for example, has two separate chromosomes. And they're both essential, so if I try to remove one chromosome or the other, and they're both circular, that bacteria would not survive. So there's essential genes on both chromosomes. And as we'll see with a lot of examples coming down the pipeline, the next number of lectures, is that there are plasmids in a lot of pathogens and in bacteria in general. And so there are some bacteria that have no plasmids. There are some bacteria that have one plasmid. There's some that have tens of plasmids. And they're just essentially, remember, they're small circular, autonomously replicating DNA elements that are sometimes transmissible, sometimes they're not. They're essentially heritable, just like the chromosome. And when we think about bacterial chromosomes, they range in size. And so what we see in a lot of pathogens that we can isolate and grow in the lab is that these free-living pathogens, so say Salmonella or pathogenic E. coli, or a good number of the pathogens that you guys have chosen are culturable, that we consider these free-living, right? And that they have the necessary and appropriate functions, that they don't need a lot to grow and survive. They need a good source of carbon, maybe a source of nitrogen, phosphorus, sulfur, and some trace elements, and they'll generally be okay. And so what the sort of size limit for this is right around 1 million base pairs in size. And if we kind of take that 1 million base pairs in size and say each gene, and I'll talk about that in a second, is about, on average, 1,000 base pairs in size, you know, you're looking at like 10,000 or so. All right. Yes. So I'll say like around like 10,000 genes or so. And the constellation of all of these genes are all the metabolic pathways, sort of molecular dogma, structural genes that are for replication, transcription, translation, and a number of other host functions, signal transduction, all kinds of stuff that's generally required for living on your own. Now, once we get below this kind of interesting speed limit for being free living or parasitic or symbiotic, once you're below this number, about around 1 million base pairs, you become restricted in what you can actually do. And so as we kind of winnow these genomes down to obligate human pathogens like Mycoplasma genitalium, it's a urogenital pathogen. And so this particular pathogen, its genome is 580,000 base pairs. It has predominantly functions, the genes that encode functions for DNA replication, transcription, translation, a little bit of signal transduction, and basically like enough metabolic enzymes to make enough constituent components of the cell membrane, some nucleotides, some amino acids. It essentially relies on us to provide everything else. So all of the trace nutrients, a large number of amino acids, certain nucleotides, all kinds of things have to be provided by the host. So we have this interesting kind of metabolic relationship with pathogens like this. And so studying these parasitic pathogens is actually, because of these relationships, really difficult to do. And so the genomes, or the chromosomes of bacteria, they need to be packaged. So those, say, 4 million base pairs of E. coli chromosome, they need to get smushed into a very, very tiny volume. And this occurs through supercoiling. So essentially, if you think about, I didn't bring my piece of yarn today for this, but if you would twist a string and put torsional stress on that string, eventually that string is going to start wrapping around itself. If you put enough torsional stress on it, it'll start wrapping around itself even more. That's essentially what's going on with bacterial DNA, and how it remains in these highly supercoiled states is that there are DNA binding proteins that essentially are kind of like, they serve a histone-like function. I'm not going to even say that they're histone-like, but that they are nucleoid proteins that essentially help pack this DNA down into a tiny, tiny volume that fits into something that is femtoliters in volume. So we're talking tiny, 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 tiny amounts of volume that hold. If I were to stretch out the E. coli genome, it's like a half meter or something in size. So you're smushing all of that into something that you can't see with your naked eye. So that's one of the important features of these genomes, and that they are to some degree constrained in size based on the physical size of a bacterium, and essentially, say 10,000 or 10 million base pairs is one of the upper limits that you'll see for bacteria in terms of gene size. And so within all of these genomes are a large number of genes. So when we talk about genes in prokaryotes, much like their eukaryotic analogs, there needs to be a signal to tell that gene to be transcribed from DNA to RNA, which ultimately gets translated to make various proteins needed in the cell. There's a 5' untranslated region, and for bacterial MR, what this does is transcription begins at the promoter, transcribes through to a terminator, and there's a coding sequence, which is essentially what will ultimately be turned into a protein. And in this 5' untranslated region, or 5' UTR, in bacterial mRNAs, there are signal sequences to instruct bacterial ribosome where to begin translation of specific reading frames in a gene. And so this concept of what a gene is becomes a bit convoluted for bacterial systems or bacteria because of formation of what are known as operons. And so the eukaryotic gene, there's essentially this structure for like a single open reading frame. This has a single promoter and a single terminator in most cases. Now, bacterial mRNAs do not undergo differential splicing or splicing in general. Now, what happens for most genes or most open reading frames or coding regions in bacteria is that they are assembled into polycistronic messages, which are known as operons. And what this means is that you have multiple open reading frames that are co-transcribed under the control of a single promoter. And then these mRNAs that are produced as a result of having these open reading frames stacked on top of each other, they are translated simultaneously. So this allows for really exquisite control over, say, metabolic pathway induction. So if you have a multi-stage pathway for something, where you have multiple parts to put together, say, a type 3 secretion system, for example. If you package all of those genes together, and they all need to get turned on under a certain specific set of conditions, bacteria can use one single promoter to drive the expression of multiple protein components in a particular process. And so, in general, we find this to be true, that most open reading frames in bacteria are assembled in operons. So most mRNAs you will find in bacteria contain multiple open reading frames. There's not a lot of kind of singleton genes out there for bacteria. The vast majority are packaged in this operon framework. And so is any of this foreign to anyone right now? It's okay if it is. Okay. So I'm hopefully treading over kind of well-trodden earth for you guys. Now, the other concept that becomes important to think about is that there are operons. And so beyond operons, there are what are known as regulons in prokaryotic systems. And what these are oftentimes a particular process has multiple constituent parts, right? And so I'm giving you the example of, so the lac operon, so facilitating lac. Well, good thing that's water. Okay, and we're on the ground floor. So, all right. Enough clumsiness. So, the lac operon, the genes encoding it facilitate lactose transport, lactose utilization. E. coli, all of those genes are packaged together in an operon, and that is sort of a one-trick pony kind of system, in that it's co-regulated, and you can switch it on and off. Now, utilization of a sugar like maltose requires a lot more sophisticated integration into central carbon metabolism, requires more enzymes, more control. different types of transporters. And so what we find is that there's often a central regulatory system controlling multiple parts of a particular pathway. And so for maltose, for example, utilization in E. coli, there are sort of 3 islands of genes, and we'll kind of get to what this idea of islands means in terms of genes on a chromosome. But you often find, again, these sort of packaged operons that are co-localized in regions on the chromosome, and that what these regulons do is sort of one step above controlling a single operon. If I need to control an entire complex pathway to do something, I can have one central regulator or multiple kind of central regulators that control multiple operons simultaneously. And so you actually inadvertently in microbiology and genetics saw an example of one of these central regulatory hubs in the control of the lactose output. So cyclic AMP receptor protein, or CRP, or cyclic AMP activator protein, it was called CAP. They're synonymous, but under glucose limitation in most bacteria, they have a CRP analog that senses increased levels of cyclic AMP, which is a starvation signal for bacteria. And it was also a starvation signal for eukaryotes, right? So it's one of these universal signaling currencies throughout life, telling us it's a very, very old signal, right? So, nutrient sensing and being able to say, I'm in a nutrient-limited environment or I'm in a glucose-limited environment, and you're producing cyclic AMP. So this maltose regulon is also controlled by CRP, as is the lac operon. And so you can think of all of the genes that CRP controls as a CRP regulon. So it controls hundreds, and arguably, there's some arguments, thousands of genes in E. coli and its relatives, and in the bacterial world in general, that have these cyclic AMP receptor proteins. So now it might be attractive to think, all right, why aren't all of these genes just all packaged together in one place on the chromosome? What we're going to, there's a lot of interesting arguments for why this might not be occurring. And so for things that are sort of permanent fixtures of the E. coli genome, so this maltose regulon, there's some interesting evidence that in terms of 3-dimensional spatial structure inside of a bacterial cell, that we kind of think of, it's just like, all right, this genome needs to fit in this tiny sack. It's like, if any of you guys have been camping and you have like a sleeping bag that's sort of puffed up over the last couple of days, and you're like, all right, well, it's Sunday afternoon, time to go home. You get out this bag that somehow your sleeping bag fit in before, and you would jam that sleeping bag into that bag. It's sort of the same thing that's going on for the bacteria in terms of maintaining chromosome inside of the cell. And you might think of like, all right, every time I stuff my sleeping bag back into this sack, I start with where my feet are or where my head is. And I start from there and I kind of push everything together. And if that is true, there's some evidence that there's not necessarily randomness in how the chromosome is packed inside an E. coli cell. And that there may indeed be some 3-dimensional spatial organization that these maltose genes, for instance, in 3-dimensional space might actually be very, very close to each other in terms of how the E. coli genome is packaged inside of the cell. So there's interesting evidence to support this. There's, I don't know, a wonderful direct experiment, but there's some interesting examples that if I say move or invert or move the location of different pieces of the chromosome throughout the E. coli chromosome, that certain regulatory systems don't, or certain regulons don't work as well as they would if they were in a normal spatial location on the genome. All this said is that we're going to see something very different and very interesting when we start talking about pathogens and these genes associated with pathogenesis today in Thursday's lecture. So keeping in mind that you have these packages of genes that are all kind of localized throughout the genome. So in terms of organization, we're going to see again in the next lecture is that there's this sort of systematic organization of structure in prokaryotic genomes, and you have genes, and that these coding regions often are packaged together in operons, and that these operons are often components of much larger regulatory networks known as regulons. And one of the fates of what we're going to see has to do with how well are these things transmitted in terms of horizontal transfer. And that's going to be the topic of the next lecture. And we're going to get some insights into how do we get a picture of what a pathogen's genome looks like. And then we're going to begin to ask the question of, well, how did a pathogen become a pathogen? Right? So I alluded to this very early on, that many pathogens, their origins are commensals. Right? So we have E. coli that lives in our colon, and we like that it's there. It does really nice, wonderful things for us in terms of digestive health, competitive exclusion of other pathogens, but there are also some really nasty pathogenic relatives of those same E. coli. And so what makes a pathogen a pathogen? And that's kind of what we're going after in the next few lectures. Okay, now to take things even further, and we'll get back to this at the end of today's lecture, is that there is this idea that you saw a first glimpse of this, that Salmonella has radiated in terms of its evolution. And a lot of this is driven by host association, spatial segregation, all kinds of stuff has sort of driven Salmonella to diverge as a species, and that there are a lot of different subspecies. Now, if I were to go and look at the genomes of what's in all of the genomes of all of these differentiated subspecies of Salmonella, I would find wild differences in all of them. And we'll kind of get to some of this today and tomorrow, or on Thursday. But if I compared all of them, I could say, all right, if I'm going to make a tabular list of every single gene that's present in every single subspecies of Salmonella, I could find there are core components that I find these in every single isolate. So if I look down the column of my tables and I sorted things and I said, all right, they all have this DNA polymerase, and they all have these ribosomal proteins, and they all have these specific, biosynthetic pathways where they don't have certain things that collectively compared to other things. I would say that these are core genes. So they're found in all members of the species, regardless of where I found them, and regardless of who they're associated with normally. Then there's an interesting concept of what we call the pangenome. And the pangenome, you can sort of think of as, like, I don't often like making religious examples, but if we were to say there are some core tenets of most religions, right? And that they're all sort of fundamentally based on a set of core principles. And then beyond that, where this religion is practiced throughout the world, for example, it adopts the flavor of wherever it is practiced, right? And that there are components that are not found in all of the species, for example. Or we could think about this in terms of food, right? That, like, I would argue just about every culture on Earth makes bread of some kind, right? And so what that bread looks like is very, very different all over the world, right? And so what drives the diversity of bacteria and bacterial pathogens is what's in its pangenome. All the genes that are not found in every single species. And these are often associated with what we term mobile DNA. And that these are pieces of large components of DNA that move in and out of bacterial lineages. And so some of you guys are going to have the fun of talking about this in what we know about particular pathogens, because some of you guys have chosen some pretty interesting examples of really us being able to drive these concepts of there being a core genome and there being a pan-genome. And so the pan-genome you'll often find comprised of things like plasmids or transposons, which are essentially chunks of DNA that are able to move around in bacterial chromosomes and be lost and gained. Integrated phages, so viral infections that become prolonged, stable, dormant infections in bacterial pathogens. And so we'll get into how this drives a pathogen becoming a pathogen. But importantly, there's sort of the core of what makes this bacterium that particular species, and there's all of the stuff that sort of fluidly moves in and out of this genetic lineage through time. So are there questions about this concept? So I know when I first learned this was but anyway, so we'd essentially begun sequencing a bunch of E. coli, and just being able to compare your lab rat E. coli versus a urogenital E. coli, so it causes urinary tract infections, versus a hemorrhagic E. coli. Is anyone old enough to remember Jack in the Box E. coli? Yeah. Probably the only ones. Okay. Anyway, there was I think I have an example. I talked about this, but there was a pathogenic E. coli that is a commensal to cows, but essentially is a really nasty pathogen to us. There was some contaminated hamburger, and it was distributed to Jack in the Box restaurants, and so for a long time, Jack in the Box had a horrible reputation. Somehow survived as a franchise. But when we were able to compare all of these E. colis, we were able to understand some of the really fundamental differences between our lab rat, which was isolated from someone's poop 100 years ago, and what made this particular pathogen so nasty that it killed people after eating hamburgers, right? And so that all had to do with what was in this pan-gene. Okay, so how do we study this stuff? And so this is hopefully, a couple of these are going to rehash, hopefully genetics, but there are a large number of approaches for studying prokaryotic genomes. So the oldest and most tried and true still is employed in laboratories around the world, like my own, are genetic approaches. So mapping function and behavior to specific genes. Okay, and so this requires you being able to mutate genes and look for changes of function, or predict that a gene enables some function, and if you mess with it, that you can map this particular gene to a particular behavior or function. So in the cases that are pertinent to us in this course are things like producing toxins, making adhesins, doing all kinds of stuff that makes a pathogen a pathogen. And we can also now engage in DNA sequencing. And so in the traditional sense, this was the kind of laborious, sequencing of regions, or in a few cases, entire prokaryotic genomes, identifying coding regions, and this enabled the power of comparative biology in the prokaryotic world. So being able to say, all right, we have this E. coli, we have this E. coli. Are they different? Are they the same? How different are they? What's different about them? What do they do that's different? And when you build up a large enough database of known genes and known functions, when you find something new, you can say, all right, I'm going to say, does this gene that I found look like anything else anyone else has ever found? And so if you've ever used BLAST, for example, then course, this is enabled because of DNA sequencing. And then there are what I would argue are now next generation approaches towards understanding the sort of link between genes and function and what genes are present in a particular organism and what they can do. And we'll go through some of these approaches. So transposons and CNV. So in and amongst these genetic approaches, there are uncreatedly, there's forward genetics and reverse genetics. So forward genetics is you have something that's observable. This pathogen causes diarrhea. And you are going to sort of hunt for genes that, through mutational analysis, through any number of approaches, I'm going to try and correlate a bunch of genes with diarrhea in terms of this pathogen. Or I might engage in reverse genetics, which is essentially you have genes that you believe are involved in a particular phenotype, causing diarrhea in a host. And if I go and knock that gene out in that pathogen, and it stops diarrhea from happening in an animal model, then I can say, all right, this gene has something to do with causing diarrhea in the host. And so you can, I think, begin to see that the forward genetics approach is when you don't know, frankly, a damn thing. Right? If you know that, all right, Koch's postulates told me that this pathogen causes this disease, and I want to know what genes are involved in causing that disease, so I'm going to engage in some forward genetics. Reverse genetics are things like, hey, I have this new salmonella, and I've studied the heck out of an old subspecies of salmonella. I sequenced large parts of this new salmonella's genome, and I know that it has these different things compared to these other things. So in our last example, cytolethal descending toxin. So the salmonella that I work with in my lab does not produce that. Salmonella typhi, it's associated with infected humans, does. And so I might be able to say, all right, I think this cytolethal descending toxin, I think that's pretty important for human infection. And I can knock that out and look to see what those effects might be. So that's an example of reverse genetics. So I kind of alluded to some of this in my kind of last little rant here, but forward genetics was the traditional approach, and still is in some ways, to finding genes associated with causing virulence in the host. So what genes encode functions in this pathogen's toolbox that's able to allow it to interface with the host and cause disease. And so traditional methods would include chemical mutagenesis. So if you think back to any course in genetics, you would have talked about like data. You're like looking for metabolic pathway mutants in a filamentous fungus. You're essentially you're causing elevated mutation rates to find mutants in a particular pathway or a particular process, and that this requires screening massive amounts of mutants. You can use transposon mutagenesis, and if you're lucky enough nowadays, there have been a large number of well-funded efforts to generate system-wide or genome-wide knockout libraries of particular model organisms and pathogens. So what you could do then is to say, all right, I think this is important for this particular part of the pathogenic process. I'm going to order all 8,000 knockouts from this particular library, and I'm going to enlist the help of a bunch of eager young students to come into my lab and perform this assay on 8,000 different individual knockouts of specific genes. And I can say, all right, well, there were 30 genes of these 8,000 that actually appear to be involved in this process. So you can make these processes much more systematic. And this also requires some screen, right? So you have to be able to differentiate one mutant from another or compare it to wild type bacteria. And then you have to have an observable phenotype of some kind. And so I've kind of made some examples that like, hey, virulence should be easy because this mouse is really sick, right? But the problem is if I want to take that genome-wide knockout library, which is the most systematic thing that I can now do in a lot of pathogens, I want to take all 8,000 of those individual gene knockouts, and I want to test their effect in a mouse. So if I wanted to test all 8,000 of those individual mutants, how many mice would I have to infect? Very minimal.
That many?
That many? Sorry. 8,000, right? So bare minimum, I would need to infect 8,000 mice. So has anyone seen 8,000 mice? I mean, I've been in some pretty creepy subway systems around the world, and I may have seen hundreds of mice, like thousands of mice. That's pretty incomprehensible, right? So 8,000 mice, you're just, it's not going to happen, right? So we have to play some tricks. We have to do some interesting things to try and connect these genes to virulence in animal models. And one interesting approach I will take you through is what's known as in vitro, no, it's in vivo, sorry. In vivo expression technology. And there's also sort of the modern, much more modern incarnation of this known as transposon sequencing. So the simplest, most direct method for genetics in pictorial terms is if I were to use a transposon. So chemical mutagenesis is one way you can go. In prokaryotic systems, I would say, please don't do that. Right? You're asking for a world of pain because we have transposons, and what these are genetic elements that will excise themselves from one source in its DNA source and insert them into another. Pictorially shown here. And our genomes are littered with transposons. Plant genomes are littered with transposons. All genomes of any organism, any domain, period, are littered with transposons. And so these are constantly moving around for us, in terms of how long our generation times are. We, on a very local time scale, don't see these things moving around. In terms of the arc of human history, I would argue these things are very mobile. But for bacteria, we can genetically introduce these things and search for insertion mutants that are disrupting a particular gene, basically knocking out its function, and we can begin to generate a lot of mutants by transposon insertion mutagenesis, and then I can say, all right, I'm going to pick individual colonies, and I'm going to screen on two different medias. One's complete, one is a minimal medium or selected medium, and I can find mutants that don't grow, right? And so one of the approaches I'm going to talk about, and so in terms of classic genetic screening, you have two different conditions, like one that is non-selective and a selective condition. So do you think survival inside of a mouse is a selective condition? Or is any mutant can grow there?
Selective.
It should be very selective, right? And it is. So are there thing to keep in mind is there a way that I can take all of these insertion mutants and have a condition that's permissible and a mouse. And how could I think about measuring whether or not that insertion mutant grew in the mouse or not, and whether or not it's present in this permissive group. And so that is one of the most important modern tools that we have for distinguishing what genes are involved in pathogenesis for a particular pathogen. And I'll get to this technique in just a moment. Okay, now I need to do a quick interlude into genome sequencing. So genome sequencing is, so when I began my career, this was a hot thing to do. So this was first generation sequencing. And that this is this dideoxy termination method. Big di terminators, you can get sequence reads of 500 to, I don't know, I guess you can get upwards of 3,000 bases doing this now. And this is what was by and large used for human genome project. So the first draft genome that came out in 2001, that was driven by Sanger sequencing, this dideoxy method. And there's the second generation sequencing, which was something that that emerged when I was a graduate student. It was horribly expensive to do, but there were a couple of interesting methods to essentially take this Sanger sequencing process and parallelize it. So just like we parallelize computing now, that my phone here I think has, I don't know, 8 or 16 processing cores in it, something ridiculous. That my phone can do more computing than I think every computer that I own right now. And so by parallelizing this first generation sequencing, now I can sequence a whole bunch of different things all at the same time, kind of in a very high throughput way. So doing so, you're able to sequence someone's genome in kind of one go. And this has been sort of the benchmark of what's driving all sequencing technology now, is can we sequence genomes cheaper, faster, and higher fidelity. Third generation. So what this whole these second generation sequencing platforms, what they opened the door to was, all right, you can parallelize sequencing, and not only can you sequence a whole bunch of things all at once, but you can also estimate frequencies of particular mutations in a population. So if we go back to this picture, I have on this plate, I don't know, 200 or so transposon insertion mutants. And I can painstakingly screen all 200 of those on agar plates. Or what if I just scooped them all together? Every one of those transposons is in a unique place in the chromosome. And I were to grow one group of these cells in a complete medium, one in a selective medium, and insertion sequences in the selective medium wouldn't grow, but they would grow here. And so if I could sequence the DNA from the end of this transposon out into the chromosome, I know where all of those transposons are. And based on the amount of sequencing data that I get from specific insertions, I can say that there were more of those insertions in this population than not. So does this conceptually make sense? No. Okay, if it doesn't, because I'm going to touch on it again. So where this has been the most powerful is in terms of understanding the genomes of pathogens. So what happens now, the CDC has an entire sort of division that does this. There's a pathogen outbreak of some ilk that was isolated, sent to one of the CDC facilities. They culture this thing. extract genomic DNA, they shear the DNA down to sequenceable fragments, and they essentially use these next generation sequencing platforms to sequence all of these different fragments and computationally assemble the genome of this pathogen. And we essentially have done this. So when I started in science, there were, I don't know, 10 bacterial genomes at most. Now there are incomprehensible. So there are hundreds of thousands of sequenced bacterial genomes out there. And so the amount of comparative biology you can do now is incredible. And so if you want to know a future area in biosciences is being able to effectively perform comparative biology. All right. So I'm going to give you an example of an old forward genetic screen. We may have to end after this one. And this is this in vivo expression technology. And what this particular technology relies on is that, if you remember way back to one of the first lectures, I gave you this sort of molecular Koch's postulates. So one of the important contributions by the person who came up with them is this idea that virulence traits are not active all the time, right? That they are induced under specific conditions to enable that particular function in that pathogen. So what you can do is you can actually exploit that fact very cleverly. And what you can do if your pathogen is genetically tractable is that you can generate auxotrophic mutants that otherwise will not grow and be pathogenic in the mouse. And so there's this example of pure A here, so you're not able to make purine nucleotides if you have this mutation. So if I were to take a pure A mutant of Salmonella, throw it into a mouse, the mouse will be completely fine. That mutant of Salmonella will not make a mouse sick. Now what I can do is I can create a fragment library of my pathogen's genome, and I can clone essentially every single promoter of every single operon of that entire pathogen into a plasmid vector that promoter will drive the expression of pure A. So essentially it will complement pure A if that gene is active inside of the mouse. So what you then do is introduce these plasmids into your pure A host, and then these 1 through 4 here are just iterations of this same idea that you'll find out there. And if this particular promoter is turned on in the mouse, you'll complement pure A. The Salmonella that otherwise would not make purines will make purine nucleotides and survive inside of the mouse. And so what you can do is make a large clone library, transform them into this Salmonella, pool all of those Salmonellas together, throw that whole pool of promoter clones into Salmonella, and essentially look at who survives in the mouse. And so after 3 days, after infection with this pool, I would go ahead and sacrifice these mice, pull out their intestines and spleens, grind them up, plate them out, and isolate individual colonies, and pick all of those colonies, and sequence what promoter fragments are present in these clones that survive. Because if they survived in the mouse, it means that those genes were active and turned on in the mouse. So this is going to be littered with a whole bunch of things that are what we call housekeeping genes, that they need to be expressed all the time. But when you subtract out all of those things, what you're left with are things that are arguably turned on in the mouse and turned off outside of the mouse. And so that's what this, you oftentimes have some kind of colorimetric reporter attached to the same expression system, so that inside of the mouse, this pure A gene is turned on, but the same clone outside of the mouse is, in this case, if I were using LAC-Z, would be white on X-scale plates. Okay? So what you can do is you can enrich for clones, promoter region clones that are associated with particular operons of genes associated with virulence in mice. And so it's a forward genetic approach to identify lots and lots of different genes that you can then use reverse genetics to go back and test the function of specific genes in its relationship to virulence. So are there questions about this method? Okay. So try and ruminate on this one a little bit, because I'm going to ask you some questions about it. Okay? All right. Now, back to this idea of transposon sequencing. So one of the things about next generation sequencing is if I have a mixed population of DNA, and I were to sequence all of it, the number of sequencing reads that parallel sequencing reads that I got would be proportional to how much of that particular piece of DNA was in my original mixture of DNA. So what I can do with this idea, say I wanted to know about a particular allele frequency of, say, some human-associated allele with some particular thing. I could get genomic DNA from everyone in this room, sort of PCR amplify a particular locus, sequence it, and if there were, out of all of the reads that I got, there was 1 in 1,000 of this particular allele, and I can work out that, like, all right, this is its frequency in the UMSL human population, right? So we can do the same thing with bacterial systems, and if we lump together all these transposon mutants, and we sequence where all of those transposons were before and after some treatment, what is being illustrated here are sequencing reads that align to a particular gene, and that's where a transposon landed. And so the idea here is that you begin with, say, a population of insertion mutants that you might have a number of insertions, and you're considering 2 genes, gene A and gene B. I start with an equal this is what the population of this number of insertion mutants look like. I expose them to some shift in an environment, and I see the frequency of insertions go down in gene A compared to gene B. So I might say that, all right, by comparison, gene A, I think, is more important for this particular environment, because if you lose function in gene A, you're less fit. So there's fewer of these mutants that survive after exposure of some time. So this is a fundamental shift in how we explore pathogens now, because delivering transposons into pathogens is very easy. It's really time efficient, and now I can basically take a whole massive genome-wide set of mutants, put them into an animal model, and look for what genes are important for surviving in that particular animal model of infection. And so it's actually really similar to the competition assay that I talked about a long time ago, several lectures ago, right? So we're looking at what is the fitness of a particular mutant in a new environment. So with that, I will leave you to